This video is part 1 on DNA replication, and it will contain an overview of the process of DNA replication as well as a review of DNA structure. Let's start with the review of DNA structure. Right here, you see a nucleotide. A nucleotide is made up of 3 main parts: the phosphate, the sugar, in the case of DNA, deoxyribose sugar, and the nitrogen base. The nitrogen base is the variable region of the nucleotide, and in DNA can contain the nitrogenous bases A, T, G, and C. When we look at a DNA molecule, we'll notice that the 2 sides don't look exactly the same. That's because of the directionality of DNA. As we recall from our nucleotide structure, the top of the sugar contains an oxygen, and then each of these intersecting points represents a carbon. So this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Carbon 5 has a phosphate attached to it when we're at a single nucleotide. So if we look at this DNA structure right here, we'll notice that it has 2 strands of DNA. We often call this structure a double helix. The sides of the DNA are made up of sugars and phosphates. And the steps or rungs of the DNA ladder are made up of nitrogen base pairs. Okay. If we think about base pairing, we'll remember that a purine, which is a 2-ring structure, always pairs with a pyrimidine, which is a single ring structure. So in the case of DNA, A pairs with T using 2 hydrogen bonds to hold them together in the middle, and G pairs with C using 3 hydrogen bonds to hold them together in the middle. And again, we can talk about the directionality of the DNA. On this strand of the DNA, we'll see a sugar with a phosphate attached to it. So that's carbon 1, 2, 3, 4, 5, carbon 5. So this is the 5' end of this DNA strand. If we go all the way to the end, we'll see the 3' carbon sticking out. So this is the 3' end of this DNA strand. Now if we look at the second DNA strand down here, we see a phosphate sticking out of this side of the DNA molecule. So that's the 5' end. And the 3' carbon on this side of this DNA strand, this is the 3' end. So we can see with the 2 strands of DNA, they are moving in opposite directions in terms of the direction their nucleotides are facing. So we call these strands anti-parallel. And this will have consequences for the way that DNA replicates. Okay. And we also talk about like the directionality of this, or the idea that one of the DNA strands is a 5' to 3' strand in this direction. The other DNA strand is a 5' to 3' strand in this direction. Again, this will have consequences on how DNA replicates. Now, how does DNA power, how does a cell power DNA replication? If you recall, when we were talking about ATP, we talked about how ATP is a modified nucleotide. That's going to come up again in this unit. Because if we look at this nucleotide, this is called a nucleoside triphosphate. It's very similar to the structure of ATP, right? ATP is like adenosine with 3 phosphates. Here we have the sugar and the nitrogenous base with 3 phosphates. But you could have GTP or TTP or CTP. The bonds between these high energy phosphates are going to be what provide the energy for DNA replication to occur. Okay, for the bond, basically for the bonds to form between the nucleotides. So I always think of the nucleoside triphosphate as a battery pack. So we have our battery pack, and how does it work? When we want to join this nucleotide to a DNA strand that's already forming, we can break off these 2 extra phosphates, and the energy released can be used to form the new bonds between this nucleotide and other nucleotides. So if we break the high energy bonds between the phosphates, the energy released can be used to form new bonds between nucleotides. So this basically powers DNA replication. As a consequence, however, if we take one of these nucleotide triphosphates, we add here's phosphate, 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 here's the rest, here's the sugar, here's the nitrogen base. This is a nucleoside triphosphate right here. If we think about these two ends of the DNA, this nucleotide is going to join to the 3' end because we need a phosphate to join to a sugar. This nucleoside triphosphate can't join to the 5' end because there's already a phosphate sticking out there because the bonds form between the phosphates and sugars. So what's going to happen is as this power pack is broken off, as these extra phosphates are broken off, a bond will form between this exposed 3' end of this carbon, of this sugar, and this phosphate. But that means that DNA can only grow off of the 3' ends because of the way that the bond formation is powered. So before we get in depth into the steps of DNA replication, I'd like to briefly go over kind of an overview of the process of DNA replication. First of all, the whole point of DNA replication is to copy DNA so new cells have identical DNA. Okay. We also know that DNA replication is semi-conservative. So if we have a DNA molecule, and we want to replicate it, we know that that DNA molecule opens up and each original strand of DNA, so here's the original DNA, each original strand of DNA acts as a template for new DNA nucleotides to be added. So here's my new DNA nucleotides. So at the end of this process of DNA replication, each DNA molecule is made up of one original strand of DNA and one new strand of DNA, but the end result is the same. The result should be 2 identical DNA molecules. So if your cells are undergoing the process of mitosis, each new cell will have identical DNA in it. Okay, right, the blue here is the new DNA. Okay. Another important part of the overview of DNA replication is that complementary base pairing occurs. Which of course A means A pairs with T and G pairs with C. And this helps with the continuity of life because we know that if DNA opens up and there you see a code ATG, complementary nucleotides will come from the nucleoplasm to make the correct new matching strand. The last thing that we know is that DNA only grows in the 3' direction because of the nucleoside triphosphate. In the next video, we'll look more in depth at how DNA replication actually occurs, what the enzymes are, and what they do.